about the antibiotic resistance in Helicobacter pylori. So let's start this session, and it's a pleasure to introduce Professor Butler, just for you to from your CV.
Not too much, please.
No, very short. And Professor Butler is full professor at the University College Dublin at the School of Biosystem and Food Engineering. He previously worked in production management in the Irish dairy industry, so he's an expert on cheese production. And his main research interests are in food safety, with a particular focus on food chain integrity and quantitative risk assessment of microbiological and chemical hazards in foods, and increasingly using a next-generation sequencing technique and data analytics to identify the sources and routes of transmission of pathogens to the food chain to control their occurrence in foods. So, please, Francis, the stage is yours.
Good, thank you for that introduction.
So you have the pointer here, you have a mouse for the
Perhaps you can
slides,
follow from.
and you have a screen.
Okay, good. And we are live. Good. Which one is the pointer? This one. Perfect. Okay. Well, thank you for those very kind words. Actually, I'm going to set a timer so that I will not hold you too long this morning. It's the worst thing to have to listen to a long, boring presentation, so hopefully I will keep it relatively short and hopefully a little bit interesting. As the introduction says, I am actually, from Centre for Food Safety at the University College Dublin, but in background I am engineer, so I am not a microbiologist. I am going to talk about, the subject area will be microbiologist, but my background is more risk assessment, and a very nice, I would say, very fruitful collaboration between myself as engineer, who is doing a little bit the mathematics, and microbiologist who is doing the laboratory. So please do not never let me near a laboratory. I would make such a mess, but I am doing a little bit then what are the results from the laboratory telling us. My title, I will talk a little about thermal inactivation for several reasons. They will come apparent as I go through my presentation. And I've changed a little bit the title. I've taken a little bit from the title of the workshop this morning, which is New Challenges, but really, I suppose I'm going to talk about old challenges. It's what I will talk about, in a sense, is actually nothing new. We have known about thermal processing and thermal validation and the rules for quite some time. But I think what is new and what is challenging is what the industry is doing. And I think if you go into a supermarket, a retail outlet, I think the speed of change in what we are seeing in supermarkets is, behind the scenes, is quite amazing. Particularly in the area of ready to eat or ready to consume. Maybe you bring it back and you must heat it, but the products we are no longer cooking in our kitchens. We are basically relying on somebody else to do the cooking, and then we as consumers, we either eat straight away or in the microwave. And that to me is creating huge challenges in terms, particularly for Listeria, because it is potentially providing an ideal opportunity for this pathogen to grow and to cause illness. We'll try success. Okay, so a little bit of this is actually coming from, it is not my first time here in Teramo. We have had quite a nice collaboration in the first instance with our colleagues who are here this morning from IZS, but also through IZS actually back to the university here, to Maria Shona, in terms of PhD supervision. So it has been quite a nice collaboration where we were looking at, the title is there, kind of novel predictive microbiology, which I suppose is my background, as well as risk assessment. And in that project, let me try this one. Yeah, perfect. We had four, actually, project which was funded by the EFSA, European Food Safety Authority. We had 4 partners. I was the coordinator for this consortium. We had partners, the Institute Polytechnic de Braganza from Portugal, IZS here from Teramo, and also University of Cordoba. And it was, I think the important word there was capacity building. I mean, we did some research, but mainly it was a distribution, bringing together all our knowledge, and hopefully by adding together, all of us were gaining from this project. And again, it was looking at listeria, and particularly in ready-to-eat foods. And a little bit, again, why I want to talk today about thermal inactivation, I think we must go back and, as you pointed out, we continue to have food safety issues continue to be, a real issue everywhere globally. And I picked this one out because, well, it is with listeria, and also it was here in Italy. And in a sense, it was partly a breakdown through thermal processing in terms of the consumption of a food product, which, my understanding, maybe it's not full understanding of what has happened in this event, but that the product did become contaminated with listeria. We were expecting the consumers maybe then to heat treat, to cook the product, but not always was the product being properly cooked in the home. And it did then cause illness, and in fact, unfortunately, caused fatalities in this event. So behind this really is thermal processing, thermal validation. We have to make sure that our products we put out are safe. And like the challenge with Listeria monocytogenes is, well, it is everywhere, potentially. So we as manufacturers, we as processors, we must ensure, assume at least, that it may be present in our raw materials, but what can we do in terms of ensuring then that in the final product it is free from listeria? And the other challenge coming from listeria is its ability to be cold tolerant, that it will survive and multiply even at relatively low temperatures, at refrigeration temperatures. So this whole food sector, which is ready to eat, chilled foods, really it is ideal for the potential propagation of listeria, and we need to realize that. I think, if you come at this from HACCP, Hazard Analysis Critical Control Points, we have a number of key steps in our processing steps to combat listeria. Probably the main one is in our processing step, if we have a heat treatment, that the heat treatment is adequate to ensure the inactivation of listeria. And really, I suppose that's what I will talk about. But again, there are a couple of other steps as well that potentially are critical control points. Potentially the rapid cooling of the product after the cooking. And I suppose I should add in there really, and particularly this comes back to some of the ready-to-eat foods or the ready-to-reheat foods we see. There continues to be quite a lot, particularly in the deposition. If we have a product, maybe it's a meat product with some vegetables, it is in a package, and then the instructions are for the consumer to heat this later on in a microwave. The actual distribution, the depositing of these products, the meat, the vegetables, maybe it's rice, a lot of this continues to be quite manual. So there is quite a lot of potential for post-process contamination in our products. And again, we have to be very careful. So hence the cooling and the subsequent storage distribution, that really we have to think about post-process contamination as well as contamination that potentially survives the heat treatment. So I will talk today about the processing side, the heat treatment side, but again to remember the other side as well. Now that is, classic growth rate. It's really the spectrum for what listeria will grow. Apologies, the screen maybe is a bit but I took it from this website, actually quite an interesting, one of these so-called shiny apps, shinies that are available now, which gives us what is the growth spectrum for listeria, and you can see down here at the, basically right down to 0 degrees C, so the x-axis is temperature, the y-axis is the growth rate. So, okay, the growth rate is diminished, but it still allows it to grow down here. But really up here then at the other spectrum, at about 45 degrees, it's finished. And really above that, we start to move into an area where essentially we are having inactivation. And really this, I'm hoping, certainly anybody from a microbiology background in the audience, this is old stuff in terms of thermal inactivation, that typically we see that when we, something like listeria at a particular temperature, I think actually this is at 60 degrees C. Basically, the longer we leave it at 60 degrees C, the more microorganisms are inactivated. And generally, these are classically are fairly linear. Certainly, if you plot the log of the bacteria numbers versus time, we typically see a reasonably linear process now. There is some variation on that, and I might say something about that at the end. And as a tool, I mean, the Combase is quite a good tool to allow us to do these simulations. And typically then, and again, I mean, this is, I think, old knowledge to everybody here in the audience, is that as we increase the temperature, then the speed of reduction becomes more rapid. And from a mathematical perspective, we need some measure of this, and basically we look at the, more or less at the slope of these lines here, and we come up with this value, which we call the so-called D value, which again, I'm hoping that you have some knowledge, maybe from an undergraduate lecture some time ago, about what the, and D is the time required then to give us a 1 log reduction. So, no, I mean, today is not about the maths, but it's just really the concept that we can start to quantify what will be our reduction. And then D itself will change with temperature. And typically, as the temperature increases, D will decrease. And again, it's, I mean, experimentally, we can derive these curves. This is actually taken just from Combase. And we can see then, as I said, as we go, in this case, from 60 degrees to 68 degrees, the D value is changing, and changing quite dramatically. And actually, it is one of the practical challenges we see in the food industry. And it's kind of alluded to here by the rapid change in D value, is that for whatever reason, if we undercook, I mean, and that can happen, the steam that is driving our cooking process, there might be a problem with the steam. So it is actually very sensitive to temperature change, that even a small reduction in our temperature below the desired temperature actually can have very catastrophic effects in terms of moving the product from being a safe product to being an unsafe, potentially unsafe. So the kind of clues to that are in exactly how the D value is changing very dramatically as we go from 60 degrees to 68 degrees, which is not much of a temperature change. And then again, I mean, if we look at, we plot those values versus temperature now, we can see, well, if we just do it linearly, it's not a curve, but I mean, there's no fundamental rule to it, but conveniently, if we plot the log of that D value versus temperature, again, we get a straight line. And again, it allows us to introduce this second value that we torture undergraduate students with, which is our Z values, which if we, again, is the temperature required, the increase in temperature to achieve a 10-fold reduction in the D value. So again, it's just a little bit of mathematics, but the basic take-home message from this morning really is that armed with knowledge about our D values, armed with knowledge about our Z values, which we can do experimentally, relatively straightforward, it allows us to do this. It allows us then to look at our thermal process and make an estimation, is this thermal process actually delivering in terms of, is it safe or is it not safe? Because the reality is, I mean, these values here, these graphs, if I go back, I mean, it's very convenient. They are at 60 degrees, at 64 degrees, at 68 degrees. So our experiments tend to be at constant temperatures. but our processes typically are varying. I mean, it's a cooking cycle. I think I have a plot of it here. Well, I'll go up a few. No, I mean, typically our cooking processes look like this. So it starts to be a little bit more complicated to make the mathematical estimation. Is this kind of a temperature profile? It's not a true one, well, it's taken from a laboratory, but it gives you the general shape that if you cook something, let's say it's a can of product, that there will be a rise in temperature, there will be a finite time to get the product up to the required temperature. We will hold it at a certain time, and then there's a cooling process. So these, to go from our D values to our Z values, knowing this information, we can start to make a quantification. Is this actually giving us a reduction or not? Okay, so I mean, where are we in terms of thermal processes? Really, they kind of broadly, very crudely, we can divide them into two processes, into pasteurization process, into sterilization processes. Sterilization basically is looking at complete inactivation. So inactivation not just of cells, but also of spores. So typically we think of sterilization, and the classic example is canning. And basically arising from canning, we end up with a product that has a very long shelf life, I mean maybe 1 year, 2 years, and importantly it is ambient stable. So I'm not going to talk about sterilization today, but I want to home in, as I said at the start, to some of the processes whereby we are delivering these ready to eat or ready to cook products. So they are typically in the other category, which is pasteurization. So if we have a thermal process that doesn't basically achieve complete inactivation of the organisms that may be present, then we are really in a pasteurization type environment. And again, we divide it broadly into two categories. In both of these cases, typically we need refrigeration. So as I said, these kind of meals that we are becoming more and more familiar with, typically they are in chilled cabinets in the supermarket. So they need refrigeration to be, to partly ensure the safety of these products. And we basically divide them into what I call short shelf life and longer shelf life. And the division here is about 10 days. So a lot of these ready to eat, ready to cook meals nowadays, what you see typically are in the top box here in the refrigerated short shelf life. And as I said, really what I want to talk about for the rest of the presentation. What is the differentiation? Well, actually the differentiation, as will come clear in a moment, is our focus changes in terms of our food safety focus. In terms of, so for refrigerated short shelf life products, we are typically concerned about listeria. That is probably the single most important challenge we have for these products. Whereas if we attempt to go much beyond 10 days, 20 days, 30 days, then actually our focuses should change to some of the Clostridia, because they then have a potential, given the longer shelf life, to basically, if there's spores present, they might, waken up basically, start to produce toxin and potentially produce an outbreak or a non-safe product. So I'm going to talk about the first category here, refrigerated short shelf life, up to about 10 days. And then generally, what our pasteurization process delivers is the inactivation of any of the pathogens that are present as cells. So things like your listeria, your salmonella, your pathogenic E. coli. It is not sufficient to really inactivate any of the spores that are present. And that's why I'm making this distinction between short shelf life and long shelf life. And really what we do then in terms of defining our cooking process is we have to define it in terms of the most heat-resistant, non-spore-forming pathogen, which generally is listeria. And typically these processes are quite conservative in terms of what we say is we want to get a 6-fold reduction in it, so reduction by 10 to the 6. And this is then where our knowledge of D values, Z values come into play, because if at 70 degrees, and typically our 70 degrees is kind of the reference temperature we use for these type of cooking processes, the D value for listeria at 70 degrees C is 0.13 of a minute. So remember, a D value will give us 1 log reduction. If we want 6 log reduction, then we multiply by 6. 6 multiplied by 0.13 is 0.78. And actually, we are typically very conservative. So we say, okay, we don't just do 0.78, we bring it up to 2 minutes, which then gives us essentially a kind of a definition of what is the minimum heating required to consider the product to be safe. So which is typically, and we declare it as a p-value, it's p-value at 70, hence this of 2 minutes. So if we cook for 2 minutes at 70 degrees C, we are reasonably assured, sorry, we're not reasonably assured, we are assured that the product will be safe from a listeria perspective. And really then our Z values will allow us, so if 1 minute at 70, then we can do the calculations, we don't need to concern it, but you can see the direction that if our cooking is at a lower temperature for whatever reason, then it's going to take longer, and this equivalence is dictated basically by D values, Z values. We don't need to talk this morning about it. But we do need to think about our process in terms of it's not just giving us 70. In fact, this case, we were doing something a bit messy in the lab. We were only getting up to 60. But we do need to consider what's happening here. What is the contribution of this portion to the lethality? What is the contribution to the cooling pace? And again, our D values, our knowledge will allow us. So kind of mathematically, it's actually an integration type process. But again, we don't need to talk about that today. But it does mean then, and we can look at, if your process is delivering something like this, is it combining with the reference, which is 2 minutes at 70? Extended shelf life, same thinking, but now we are talking about Clostridia. I'm not really going to talk about it. Same kind of calculation. And in this case, if we want to push for maybe 20, 25 days, we need to be thinking about Clostridia. And oops, I keep hitting the wrong button. And now our cooking must be much more severe. So we are talking about 10 minutes at 90 degrees if we are producing. And this is really where the confusion is coming in the industry, is inadvertently, because a lot of pressure is coming in the industry in relation to extended shelf life for logistics reasons. I mean, we don't want a product that is just 5 days shelf life. We want it to be 7 days. And then suddenly the other multiples say, well, I mean, your competitor is saying 7 days. Can you give us 9 days? So inadvertently, processors are pushing the boundaries here. And we just have to be careful. We just have to be knowledgeable about what we do. So that's really what thermal validation in a nutshell is. Well, first of all, we have to consider, what is our cooking? Typically it's in an oven, a very schematic of an oven packed with product. And typically, where is the cold spot? So generally it's down here in the corner, just like in a domestic oven. So really this is the product we should be worried about. Even within the individual product, we must be asking the question, where is the cold spot? Where will be? Now again, typically it's in the geometric middle, but not necessarily, not always, depending on your product. And then finally, the part of the thermal validation is the little bit of maths. It is this integration here. If I go back to this one, what is your actual cooking temperature and is it delivering a safe product? I'm not going to say anything, but fortunately, everybody hates the maths. That has been my experience as an engineer working with microbiologists for more than 25 years now. Microbiologists hate the maths, so fortunately, there is increasing online resources to do this for us. So I'm not going to say much. I'm conscious a little bit of my time, but they are good and they give you, this is an example of, it's a very crude example. I was just doodling last night in the hotel, kind of a triangle of a cooking. What will be the inactivation then? And this will be typically the outcome. So, Chair, I think I'm kind of finished. So really just in summary, it is important. It's kind of behind. It's old science, actually. D values, Z values, they have been around for for a very long time. I think what's novel here is the products, the processes and the challenges. And as I said, the types of logistical challenges that we are seeing. Please give us longer shelf life because it aids in the distribution. We can have the product longer on the supermarket. We can sell more product because of that. And sometimes we are crossing thresholds here, or the producer is crossing thresholds that we just need to be aware of. So I will finish there. Again, just to acknowledge to EFSA, as I said, this is old thinking, but some of the data behind what we are talking this morning was generated by this project, particularly by our colleagues from IZS. And it's an ongoing, just to keep, particularly for novel products, what is the relevant criteria, the relevant growth data to support our calculations. So, Chair, I'll finish there. And as I said, it's old science meeting new products, maybe. That was the definition of my presentation.
Thank you, Professor Butler, for this interesting presentation and for giving us the idea on how it is important to be an engineer to fight against pathogens.
Well, you need everything, many disciplines.
Yes,
microbiologists, engineers.
Of course.
Even social science now, so you have to think, what is the consumer doing? And actually, I did not mention it, but also even this whole, delivery to home is enormous. So again, what is happening in these food factories that are producing the food that is then delivered to our home. So anyway,
it's a very complex matter. So we can open discussion. Are there any questions from the audience? Okay, Mauro.
I think the room is small enough, I think.
Io ti sento.
Vabbè, tanto mi senti.
Sì.
Thank you very much for the presentation. I would like to go back to the beginning, to the background.
Yes.
Because you talk about ready-to-eat food, and this is something that's happening all around the world. We are not cooking anymore. Also in Italy, it's happening that in our tradition we are used to go shopping food, choosing and cooking, but we are losing this. And behind this, apart from the important issue of the microbial infection, there is the other important issue that most of the ready-to-eat food are not very high nutritional value. Most of them are processed foods and so on. And I think this is the main problem of all this history, and also the fact that we became somehow lazy in terms of cooking. And this opened a window of eating a lot of processed foods ready to eat, then bring us to the pathology like obesity and something like that. So you say also social science is important. I agree. We need to know what the consumer wants and how to change this, because this is the main issue. How can we change the wish of consumer to grab food that is ready to eat, bring to the issue of increasing metabolic disease? I know that is not real, but just in terms of discussion.
Yeah. No, I personally agree with you. Now, if you look at an individual, I think no food is bad for you. But it's only when you start to maybe consume in excess or too frequently. So it is, it is consumer, and I think it is consumer perception, and hence I say social sciences, that we need to make this information. And how do we do that? So far we have not been successful, I think, to educate our consumers. Or educate is probably even too strong a word. It's to sort of nudge, because you're right. When you look at these processed foods on their own, they are not necessarily unhealthy, but I think they are unbalanced. So what I think what is missing in this is everything that is coming maybe from our fruits, from our vegetables, this portion of our diet, that is what is missing, I think, from these. So, I mean, if you make at home a salad and then buy in, it's okay. But if you're just the same, I mean, which is typically a lot of it, which is protein, I mean, high protein, I mean, meat-based, then yes, you certainly have issues with I mean, obesity and nutritional imbalance.
Yeah, thanks.
Okay. Thank you, Mauro. Are there other questions? Annalisa, please. Right.
Just wanting to know what's the role, well, actually, basing on this presentation, food industry could push the limits more and more
Yes.
continuously. And I wanted to know the role of consumers against this challenge. We need to be against this challenge of limits because probably quality of foods is...
No, I mean, I think there is nothing inherently wrong with producing, no, I mean, with extending shelf life. No, I mean, as long as you do it safely. But I think the challenge is for us as maybe scientists, I use that word, is to educate the food industry to understand the implications of what they are doing. I think that is the important distinction. So I think if you can manage to get 12 days versus 10 days in shelf life, that's great. I mean, it's I mean, particularly for even environmental issues, maybe it's reducing waste, lots of things. But the processor must understand, and as I said, I think that is the knowledge that is missing in the, or is not missing, but it is the industry, I think, needs guidance, more guidance in this area, particularly small industries who are maybe doing one product for many years and this product is safe. But then they say, we do something a little bit different. We use this. People want ready to eat, so we add our ingredient, make something new. And it's the novel, or it's the other ingredient that they are using, or maybe they're combining their meat product now with maybe pasta or with rice. And the problems are with the pasta and the rice, which they have no knowledge or little knowledge. So, as I said, I am not necessarily against innovation and extending shelf life, but we need to understand the consequences. No, I mean, I'm just, there was an outbreak of Clostridia in one of the Gulf states, which I have a little bit of knowledge, but it would appear to be with a mayonnaise type product. And no, I mean, mayonnaise classically is safe for Clostridia because of pH. I mean, it's typically a low pH product, so Clostridia should not grow. So that is okay if you're making, now I'm not saying that this is the cause of this outbreak, but think of mayonnaise. I mean, we've had mayonnaise for many years, but now we have many flavors of mayonnaise suddenly in our supermarkets. Some of these are no longer for instance, in glass jars that have been are ambient stable, so they have got a quite an intensive heat treatment, so they are in the chilled, they are basically again ready to eat. Maybe the pH has been changed because now they have added different ingredients, different flavorings, so maybe the pH has increased a little bit. So going from that, that threshold, which is about pH probably 4.5, suddenly they push it up inadvertently because they do not have the required knowledge. So they make a change to their process, change or increase their product range, and suddenly they have moved to kind of out of a safe space into a potentially unsafe space. Now it's only one example, but this can happen in many cases. There are plenty of examples where traditional products are safe. We move, and again, moving, for instance, low salt, we move away from a product that is inherently safe to a product that is potentially, there is conditions where pathogens can grow. So to the knowledge of you guys, it is important in the food industry. We need more of you guys to make sure, to spread the science, to understand the science.
Okay, thank you very much, Professor. The last question from Professor Annalisa Serio. Please,
Thank you,
Annalisa.
Professor Butler, for your nice presentation. And actually, I would have a lot of questions for you, but I just ask one. Do these calculation about the D value, the Z value, and all the softwares that we have to make some predictions, do they take into account the differences between different strains, or are they calculated maybe on type strains? Because you know that actually we are facing problems also because of very resistant strains.
Yeah.
So
Actually, I'm happy that you asked this question.
thank you.
Because for now, the last years, we have been looking very much at strain to strain variation in different things, partly with Listeria, partly in this project with IZS. And again, our interest has been in strain to strain variation in the kind of what I will call the industrial setting. So things like resistance to sanitizers, things like resistance to cold temperature. No, I mean, will some bacteria, again, Listeria, are there some strains that preferentially grow at low temperatures faster than others, and hence. In answer to your question, no, I mean, where is the Z, sorry, where is the D value there? D is 0.13. Yes, there is a variation there. No, I mean, a strain to strain variation in that figure. My own kind of conclusion is, see, what there is an inherent safety in what the calculations are doing here, and particularly in this step here. See, we go, I say 0.13, 6 pi is 0.78, but actually then we go from 0.78 and we say, here, not 0.78, we bring it up to 2 minutes. So really, in that kind of factor of safety, it is sufficient, in my view, to cover certainly the relatively small change in D values that we would explore, no, I mean, from strain to strain. There is definitely this 0.13 will vary strain to strain, but the factor of safety going from 0.78 to 2 minutes is actually quite enormous. And also the other thing too is, if you go back ultimately to 2 minutes at 70 degrees C, in most of our processing, we actually are doing many times that level. No, I mean, if I take an example from my own country, Irish stew, which maybe you know, or maybe you do not know, but like it is with beef, but we are cooking Irish stew for hours. So 2 minutes at 70 is nothing. I mean, so, but again, I think what is important here is to realize, and again, this is where you guys come in, is to understand what industries are we starting to approach this. So things like the shellfish industry, for instance, things like crab meat. I mean, if you cook crab meat for a long time, it's just soup, it just disappears. So actually the cooking process for quality is coming close to the safety requirements. So again, it is the knowledge, what are the critical products. So yes, I completely agree with you. And also there is some debate, it's not by any means finished. The D values typically are calculated experimentally in broth type, but the matrix could well have an input, but there is some debate whether that is more from the physics in terms of different kind of specific heat capacities. Sorry to be an engineer for a moment, in terms of the energy required, the heating. So there's some debate whether it's a true effect we are actually seeing.
Okay. Thank you very much, Professor Butler, for comprehensive reply, and also the audience for interesting questions. So thank you again, Professor. So I invite Professor Di Giulio. And Professor Di Giulio is an associate professor in microbiology at the Department of Pharmacy at the University d'Annunzio of Chieti. Her research activities focused on in vitro and ex vivo characterization of Helicobacter pylori biofilm, study the dormancy and development of innovative systems based on bioactive substances for its eradication, also in combination with drugs commonly used in therapy, evaluation of the antimicrobial, antibiofilm, antivirulence activity of innovative compounds, as an example, essential oil, semi-synthetic compounds, and so on. Please, Mara, it's up to you.
Thank you. Thank you. Good morning, everyone. Thank you, Professor, for your presentation. And I want to thank also Professor Maria Schirone for the invitation. Today, I want to talk about Helicobacter. Helicobacter pylori. Helicobacter pylori is a gram-negative bacterium, a microaerophilic bacterium, characterized by an helical shape. And it represents a highly successful pathogen that colonized near 50% of the world population. But disease occurs in only 15% of infected individuals. H. pylori infection, in fact, is generally asymptomatic, but can induce chronic gastritis, peptic ulcer, multiple lymphoma, and gastric adenocarcinoma. In fact, the International Agency of Research on Cancer has categorized H. pylori as class 1 of carcinogen, and the WHO recommend the treatment when the infection is diagnosed. Of course, the severity of infection is related to the strain's virulence and the host predisposition. The global prevalence is various in terms of age, ethnicity, in particular in the hygiene condition. In fact, the prevalence is higher in low and middle income countries such as Africa, Middle and South America, Eastern Mediterranean, and the fecal-oral and the oral routes are the most routes of transmission. Oops. As we know, the stomach is not sterile and can host its microbiome, but when present, H. pylori is the predominant bacterium. In fact, the natural habitat of this bacterium is the mucous layer just above the gastric epithelial cells. In fact, H. pylori can only survive a few minutes in the acidic stomach lumen, and it must quickly migrate in the surface of the gastric cells, in which the pH is near neutral, and this process is mediated by some its virulence factors. In fact, the key aspect of bacterial colonization is related to the production of adhesins, of course, but first of all by flagellar motility and urease activity. In fact, H. pylori, with its urease activity, facilitate the flagella motility through the mucous layer by changing the viscoelasticity of the mucus, because at low pH, the mucus is a gel that trap and block bacteria. But H. pylori, producing urease, that produce ammonium ions, rise the pH around the bacteria and change the viscoelasticity of the mucus from a gel to a viscoelastic solution through which the bacterium can swim and reach the surface of the gastric cells. Another important aspect of this bacterium is its morphology. In fact, the helical cell shape is thought to enhance, to help the bacterium to move through the viscous media, through its characteristic corkscrew-like motion. Another, of course, important aspect is the production of adenosines that can induce a more intimate association with gastric cells, of course. Regarding the pathogenesis, H. pylori can produce a lot of toxins, but the main toxins are two, in particular, Vaccae protein toxin, the vacuolating cytotoxin A, that induce, as we can see, inside the eukaryotic cells, the vacuolization, and that induce apoptotic death and necrosis in the gastric epithelial cells. Another important toxin is a Kagei protein. Kagei protein is strictly related to the neoplasia formation. Kagei, when is produced by bacteria by the type 4 secretion system, is translocated inside the eukaryotic cells. Then the eukaryotic kinases phosphorylate the protein, and in this form, Kagei can induce disruption of cell-cell junctions, loss of cell polarity in eukaryotic cells, and induce in eukaryotic cells the motility. As we can see in this photo, we can observe this particular morphology of gastric cells in numbing bird cells. The cells change morphology and start the carcinogenesis process. Beside its arsenal of virulence factors, an important aspect of this bacterium is its capability to adapt itself to several environments. In fact, H. pylori is a master of adaptation. First of all, because this bacterium is capable to evade, subvert, and manipulate our host immune system. Because this bacterium is capable to change the composition of the superficial protein and induce a deacetylation of lipopolysaccharides. Moreover, this bacterium has a small genome, and it is characterized by plasticity. Its genome is characterized also by a very high mutation and recombination rates. Why? Because H. pylori has a DNA polymerase, a characteristic DNA polymerase that lack of a classical mismatch repair pathway in combination with the promutagenic properties of this enzyme. And very, very important is that H. pylori is naturally competent. So it is capable to take up DNA and change its genome to adapt its genome to new environments, new conditions in an extremely efficient way. Another important aspect that I in particular study is its capability to adapt itself in stressful conditions, such as the presence of antibiotics or changes in environmental conditions, such as pH value or oxygen concentration. And the bacterium is capable to produce biofilm. Biofilm is an aggregation of cells embedded in extracellular matrix composed by sugars, proteins, extracellular DNA, but the biofilm represents a three-dimensional structure like a barrier against the penetration of antimicrobials. Inside this three-dimensional structure, the bacteria are capable to promote horizontal gene transfer. and protect themselves from the host immune system. Another important aspect that I love to study, yes, is the dormant state. It is a very fascinating capability of this bacterium, but it is not the only one, to produce a dormant state. In this state, bacterium, first of all, change its morphology from helical shape, become coccoid bacteria. In this form, it change the cell wall characteristic and composition. And in the coccoid form, it is very, very difficult from the host immune system to recognize the bacterium. In this form, the bacterium is viable, but not cultivable. So it is very difficult to recognize the presence of this bacterium. Another important aspect is that H. pylori in dormant state reduce urease activity, is characterized by a low metabolism, but maintain the expression of virulence factors. In these both phenotypes, bacterium is in a tolerant state and induce chronic infection, very, very difficult to treat. The treatment for H. pylori infection include a traditional triple therapy, including 2 antibiotics, among amoxicillin, clarithromycin, levofloxacin, or quinolones in general, or metronidazole, plus proton pump inhibitor for 10 or 14 days. But the problem is related to the diffusion of antibiotic-resistant strains. In fact, the W in Europe and of course in the world, the WHO in fact insert H. pylori clarithromycin resistant strains in the priority pathogen list, for which there is the urgency, the need to find a new strategy to counteract the infection. Today, there is a lot of works that include a combination with different antibiotics, but very, very interesting is the concept of drug combination. Yes, among antibiotics, but in particular among antibiotic plus no antibiotic compounds. This combination can offer a strategy to overcome this problem of antibiotic resistance. These compounds are named as antibiotic resistance breakers, compounds capable to restore the efficacy of conventional drugs and break the resistance. How? Inducing a multi-target action in the bacterium, and the bacterium is not more capable to react. For this reason, today I want to show you some of our interesting results about resveratrol, bovine lactoferrin, and vitamin C, for the treatment of chronic infection related to Helicobacter pylori. In the first work, resveratrol, we evaluated the potential role of resveratrol. Resveratrol, we know, is a natural compound, attracted great attention for its biological activities, but resveratrol presents some limitations, such as low bioavailability and low solubility in biological system. So in this study, we evaluated the antibacterial effect of resveratrol, but also we evaluated the antibacterial effect of new synthesized derivatives of resveratrol, improved in terms of solubility, in collaboration with our colleague of pharmaceutical chemistry. Then the most antibacterial derivatives were evaluated in terms of antibacterial action, antivirulence effect alone, and in combination with levofloxacin, that is one of the antibiotics that are more used in H. pylori therapy. In the end, we performed in vivo test with Galleria mellonella model. Results, they are the strain that we used in the study. They are clinical strains, and as we can see in the table, most of them are multi-drug resistant strains. Results showed that resveratrol showed a moderate antimicrobial action, but two new resveratrol number 3 and number 4 derivatives of resveratrol showed an interesting antibacterial effect, so they were selected for the other study. In fact, we tested a synergistic action together with the levofloxacin, and we can see how some of them are capable to synergize with this antibiotic. In particular, resveratrol number 3 and number 4 were capable to synergize with this antibiotic, recovering the levofloxacin efficacy when in combination of these, of course, derivatives against levofloxacin-resistant H. pylori strains. Regarding the antivirulence activity, resveratrol number 3 and number 4 alone, but also in combination with antibiotic, were capable to inhibit the biofilm formation, and they were capable to inhibit the motility of the strain. For the in vivo test, we use Galleria mellonella infection model. This is Galleria mellonella. And Galleria mellonella represents a recognized model for H. pylori infection for which no ethical approval is required. And we use this model to evaluate the toxicity of resveratrol number 3 and number 4, and the results show that they are not toxic. And this model is a good model for the infection assay. In this test, we infected Galleria mellonella with H. pylori strains. Then, after 2 hours, we injected PBS for the control, levofloxacin, resveratrol number 3, number 4, alone and in combination with the levofloxacin. And we observed a protective effect of these compounds against H. pylori infection. In this study, we evaluated the potential role of bovine lactoferrin. Bovine lactoferrin is a glycoprotein present in a lot of animal secretion, and it is an enhancer of immune system. And in this study, we performed in vitro and in vivo study. In particular, we wanted to evaluate the antibacterial action of this glycoprotein alone and in combination with levofloxacin. And in this study, we performed a pilot study. We enrolled 77 patients, H. pylori positive patients, and we divided in Group A and Group B. Group A was treated with a traditional triple therapy, and the Group B was treated with the triple therapy plus bovine lactoferrin. Bovine lactoferrin has great antimicrobial action for a lot of gram-positive, gram-negative. For Helicobacter, bovine lactoferrin was not so antimicrobial, was not so good the result, but it was capable to affect the motility of the strain, and it was also capable to synergize with the levofloxacin. In the in vivo study, bovine lactoferrin was capable to increase the eradication rate from 75% in the group A to 96% in the group B. So increase the eradication rate by 21%. Underline how bovine lactoferrin can be considered a potentiator for the restoring the susceptibility in the resistant H. pylori strains. In the last work, we evaluated the role of vitamin C in a dormant state of H. pylori. In this study, first of all, we induced dormant state following two protocol. By using a high dose of antibiotics, we induced the antibiotic persister, a form of dormant state, and in nutrient starvation we induced the viable but not cultivable VBNC state. They are two different forms of dormant state. So we incubated for the generation of antibiotic persister H. pylori in amoxicillin, but at high dose of amoxicillin, and for the VBNC state we incubate H. pylori in saline solution for 14 days. From result, for the generation of antibiotic persistent state, we need 48 hours, and 8 days for the generation of VBNC state. It was demonstrated by the changing of morphology of bacterium, zero colony on culture media, but bacterium are viable. Then the question was, is vitamin C capable to interfere in the dormant state generation? And is vitamin C able to resuscitate the dormant state? Vitamin C was capable to interfere with dormant state generation in both conditions. And as we can see, the photos is not very clear. The bacterium remain in spiral shape, and in this shape, the bacterium is more vulnerable to stress induced by nutrient starvation or the presence of high dose of antibiotics. But vitamin C was able to resuscitate only the VBNC state, but not was able to resuscitate the antibiotic persistent state. Underline how probably the pre-treatment with vitamin C in the H. pylori therapeutic scheme can can help to counteract this chronic infection. Take-home message: when we have an H. pylori infection, we have two problems: the antibiotic resistance, but also the tolerant phenotypes, because in both phenotypes, drug resistance strain and dormant state or biofilm state, it is very, very difficult to treat and kill bacterium. Today, probably novel compounds with no toxicity and high bioavailability in combination with traditional antibiotics can represent promising strategy to counteract the chronic infection related to H. pylori. We are Microbiolab. This is our website. My head, Professoressa Luigina Cellini, and nothing. Thank you for your attention.
Thank you, Mara. Thank you for a nice presentation and for this trip in the interesting world of Helicobacter pylori, very complex systems. Are there any questions from the audience? We have time for just one question. Donato, Professor Angelino.
I may miss some details about the vitamin C, for example, if one of the two isoforms is working better than other. Because, for example, with the cooking, you have not maybe.
Yes.
And the second question is, other than vitamin C, if there are some other antioxidant vitamin like vitamin E or some antioxidant that influence the.
Yes, for the isoform, probably I'm not able to answer to this question because we bought the vitamin C from Sigma. I don't remember. Yes, and for the other question, yes, also other antioxidant, in particular vitamin E. And vitamin C, I omit this particular, was capable also to resuscitate the Mycobacterium tuberculosis persistent form. And from this article, I was the idea to test the vitamin C also in H. pylori dormant form. And another important aspect is related to the fact that in H. pylori positive patients, the concentration of vitamin C is very, very low. So the addition of this vitamin could be a good solution, a good strategy to improve the eradication rate.
Okay, thank you, Mara. Just one last question from the audience. Okay, so thank you again to our speakers for their great presentations, and thank you for your attention. And now I leave the stage to Mauro. Thank you, Mauro.
Okay, so we are unbelievably in time, and probably because there was not the official presentation by the dean and the others. But don't tell to them. Okay, let's start the oral presentation of the third year PhD food science. And we start with Buccioni Francesco, that will talk about control reduction of the forte toxic and pomopsin risk by ozone treatment. Please.
Okay, good morning, everyone. It's a pleasure to start the PhD presentations today here. And, well, today my speech will be a brief snapshot of what was my PhD project. So, the control and possibly the reduction of Diaporta toxica and the related mycotoxin, that is Pomopsin A, in lupin seeds by an aqueous ozone treatment. These are the contents I will talk about during the presentation, but I suppose we can start with a brief introduction. Well, actually, greenhouse gas emissions are a real problem nowadays, and livestock sector is responsible for roughly the 14.5% of them. So in the last years, with the increase in the consciousness of consumers,
also the plant-based demand for the demand for plant-based protein sources increased, as well as the consumption of legumes that are the main source of plant-based proteins. Within legumes, lupin emerged as major players due to some of their characteristics, particularly their content in proteins. They can go up to the 42% protein content, and many facilities in their cultivations. Unfortunately, they have also some side effects, and one of them is that lupin is the main host of an endophytic, plant pathogenic, mycotoxin-producing fungal species that is Diaporte toxica, and this species is known for the production of fomopsins, in particular fomopsin A. Fomopsin A is the main cause of lupinosis, an hepatic disease of grazing animals, mainly grazing animals, that has been recently recognized by EFSA as also potentially harmful for humans. So, our approach was first to characterize the Diaporte toxica and its metabolic pathways. The second step was the evaluation of the effectiveness of some treatments. In this case, I'll talk about the aqueous ozone treatment against fomopsin A. So the first part was about the evaluation of the growth rate, the spore production, and the fomopsin A production. You can see also the alkaloid production that will be kind of surprise at the end of this part. And I'll start with the growth dynamics. We evaluated different media, both synthetic and complex media, and also the growth on lupin seeds. And what we could observe was a great adaptability of Diaporte toxica to the different media, even if in some low nutrient media the growth was lower, but it could grow on any different media. The interesting part is that we could observe the production of spore only on lupin seeds, so suggesting some need for particular conditions to multiply and to reproduce. From this point, we moved on with the evaluation of the fomopsin production, fomopsin A production. We wanted to try different inoculation procedures and different substrate conditions. So the best conditions for the fomopsin A production is the first one you can see in the slide, when we had conditions similar to the one observed during the lupin plant growth. So a water activity around 0.98 and an inoculum that spreads on lupins in a second moment to the one of the inoculation. From this result, we went on with the application of the ozone technology. Before the application of the ozone technology, we wanted to have the main scenario on the lupin situation, and we wanted to evaluate also the relationship between Diaporte toxica and alkaloids in lupin. Alkaloids, for anyone who doesn't know, are some anti-nutritional factors that are naturally present in lupin seeds. During the production process, they are removed by some debittering stages, but what we could observe, let me say surprisingly, is that Diaporte toxica is not only able to produce fomopsin A, but can produce also alkaloids similar to the ones already present in lupin seeds. So, besides everything, let's say, this must be an aspect to be considered when dealing with lupins infected by these species. Well, the second step was about the application of ozone that reacts with the functional groups of the mycotoxins changing their molecular structure and possibly producing some molecules with a lower toxicity. Well, ozone was effective against other mycotoxins, so we decided to apply it on Phomopsin A, and we evaluated it as the best strategy, also considering the production process of lupin seeds that well, ozone fits well the production process of lupin seeds. Okay, after some preliminary trials, we fixed the concentration of ozone to 7 ppm, and we only changed the time duration of the treatments. So we filled up a cubic chamber, plexical cubic chamber with water. We insert the lupins and we dissolved ozone inside the chamber. And we evaluated the growth of the Diaporte toxica, the phomopsin concentration after the treatment, and also the quantity of alkaloids by means of HPLC coupled to tandem mass spectrometry. Concerning the reduction of the fungal load, we could not really observe significant results or good results. In fact, the only significant result was after a 4-hour treatment. While the interesting part is about the reduction of Phomopsin A. In fact, in any different treatment, we observed almost roughly the 20% reduction of Phomopsin A that could be considered a good result. Concerning the alkaloids, we couldn't really identify a real pattern on the different alkaloids. We could observe a similar behavior between lupanin and multifluorine. Vice versa, on spartain, the evaluation was a bit complicated due to the really low levels of this mycotoxin, and we also observed a decrease in effectiveness at increasing treatment duration for hydroxylupanin. So to conclude, we did increase the knowledge about the Diaporte toxica that I must say is well, the literature contains very few articles and mainly outdated articles, so we produce some new knowledge about this strain. And in many cases, we demonstrated an effective of ozone, not only against the mycotoxin, but also alkaloids. But of course, before any industrial application and any scaling up of the strategy, there's the need for updating and optimize the technology fitting the production process. Thank you for the attention.
Thank you very much, Francesco. Any questions from the audience? Please. Hello?
Yes.
Also. But did you produce also some samples as controls?
Okay, in any case, yes, I'll show you briefly. Concerning the fungal load reduction, there's the relation between the control and the treatment in any different treatment. Both for the Phomopsin A and the alkaloids, I reported in the graphs the reduction percentage. I don't know if it could be... Okay. So, yes, of course. And also for...
The percentage reduction is calculated based on the control.
Yes.
Any other question? Don't be shy. Okay. Thank you very much. Let's move forward to the next speaker, and is Rossetti Alessio Pio, Enological Versatility of Starmerella bacillaris in Wine Production.
Good morning, everyone. For this brief presentation of my research project, I want to start with the aim, that was the exploration of Starmarella bacchillaris in wine fermentation. Moreover, the efforts are focused on further investigation of the effects of cell lifestyle on wine characteristic. In particular, regarding planktonic and sessile state, in which planktonic are cell-free in suspension, instead of sessile state, in which the cells are enamelled in a self-made matrix, and this state is lead with adhesion properties and biofilm formation. Some info about the background. Starmarella bacchillaris are non-Saccharomyces yeast, common associated with grapevine environment, and show interesting enological traits that put this species under the spotlight of academia research. In particular, when combined with the Saccharomyces cerevisiae in alcoholic fermentation, could allow the obtaining of wines with lower ethanol and higher glycerol content. Regarding the adhesion properties, we start from the finding in literature that microorganisms in sessile state shows a different metabolism compared to their counterparts in planktonic state. So the main question mark was how this metabolic shift could influence the characteristic of wine, and if it's possible, propose this kind of approach in winemaking production in order to obtain novel style of wine that could intercept the consumer acceptance. A brief preamble about the strain used in my work. All strain used belonging to our collection at the University of Teramo. The strain were isolated from organic grapes and must from different cultivar of Abruzzo region. All strains were previously characterized for conventional and non-conventional properties. And the strain were tested also for the ability to forming biofilm on a biotic surface. For example, here we have some Starmarella bacchillaris strain tested for the ability to adhere on polystyrene surface. We have an image obtained through the use of confocal light scan microscopy, and we can highlight that this kind of ability is a strain-dependent trait. So if we want to propose this approach, we have to take in account this information in selection and screening of our yeast and strain. Today I want to present to you 4 activities. The first 2 are related with the addition of Starmarella bacchillaris strain on oak chips. Why we selected oak chips? Because are often used in wine industry when the winemaker want to obtain wines that mimic the aging in oak barrels, but avoiding the time loss with this type of maturation. So, moreover, this type of support has a surface with pores that can be colonized by the cell that can start to grow and adhere and forming biofilm. Regarding the first activity, the aim was evaluation of the impact of Starmarella bacchillaris adhered on oak chips on the enological characteristic of and aroma profile of Montepulciano d'Abruzzo wine. So, for do that, 10 Starmarella bacchillaris strain isolated for Montepulciano d'Abruzzo grapes were used, and the strain that shows the best ability to adhere on oak surface was selected for perform lab-scale fermentation. We performed 6 trials from A to F, in which we try Saccharomyces cerevisiae, the pure inoculum of Starmarella bacchillaris, Starmarella bacchillaris adhered on oak chips, and for the last 3 trials, we performed the coinoculum in planktonic state of Starmarella bacchillaris and Saccharomyces cerevisiae, and for the trial A, we performed the coinoculum in planktonic state of Saccharomyces cerevisiae and Starmarella bacchillaris strain. For the last trial, Starmarella bacchillaris strain were previously adhered on oak chips and then used in coinoculum with Saccharomyces cerevisiae. Regarding the results obtained of aroma profile, Starmarella bacchillaris adhered to oak chips as released specific compounds such as methylvanillate, ethylisobutyrate, and ethylisovalorate. Regarding the main physicochemical characteristic, the highest value of glycerol was found in the trial F, in which Starmarella bacchillaris was previously adhered on oak chips. For the second activity related to oak chips, the aim was the investigation of potential of Starmarella bacchillaris adhered on oak chips in order to improve the enological characteristic of Trebbiano d'Abruzzo wine, so starting from a grape variety generally recognized as neutral. For do that, we perform in this case pilot scale fermentation in which we test the inoculum and the sequential inoculum of Starmarella bacchillaris and Saccharomyces cerevisiae, and the trial W5 and W6 we have Starmarella bacchillaris strain previously adhered on oak chips and then using in co-inoculation and sequential inoculum with Saccharomyces cerevisiae. Regarding the principal component analysis about the wine obtained, the trial W5 and W6 in which Starmarella bacchillaris was previously adhered on oak chips shows were well differentiated instead of the other two groups in which we have the wines fermented without chips and the wines fermented with oak chips but with Saccharomyces cerevisiae and Starmarella bacchillaris inoculated as planktonic state. For the first time, we evaluated also the difference in the color because during the buying process, it's one of the most factor that the consumer take in account when choose a wine, and we found a different B value. And in the trial in which Starmarella bacchillaris was adhered on oak chips, we have an increase of B value. Regarding the last two activity, we want to throw out the use of a support in order to obtain a more versatility technique to propose to the wine company. And for do that, we starting from the evidence in literature that cell growing as biofilm and then detached could maintain the metabolic shift. So for the last two activity, we make a comparison between the planktonic cell and the cell that we called biofilm detached cell. Regarding the third activity, the aim was the evaluation of 10 Starmarella bacchillaris strain inoculated as planktonic or biofilm detached cell in inoculum with Saccharomyces cerevisiae and chromatic characteristic of Montepulciano d'Abruzzo. The biofilm detached cell shows a wine with higher content of pyruvic acid, anthocyanin, and polyphenols content. And regarding the A value, there was an increase and decrease of B value and U angle. Interesting to show to us today is the color difference lab units, delta E, between the wines derived from the inoculation of Saccharomyces cerevisiae and Starmarella bacchillaris grows as planktonic or in the biofilm detached cell. Out of this value says to us that the value is upper than 3, that the difference in the color were detected also by human eyes. So out of these 10 trials, 6 of them shows a value upper than 3. Regarding the LAX activity, we want to make an evaluation of the influence of biofilm detached cell on the overall characteristic of Montepulciano d'Abruzzo wine. So with the same strain of activity 3, we perform pilot-scale fermentation, and the results shows quantitative and qualitative difference. In this box plot, we highlight significant difference in total amount of agar alcohol, organic acid, ester, and terpenes with a mean value higher in biofilm detached cell instead of the planktonic ones. In order to better understand the difference in aroma profile, we also evaluated analysis of other activity compound that shows a value measured than 1, and in the biofilm detached cell were characterized by compounds such as phenethyl alcohol, nonanoic acid, ethyl octanoate, and so on. In order to understand if also the panelists could perceive the difference in aroma profile, we perform a sensor analysis, and the results shows that wine obtained with biofilm detached cell were characterized by higher mouthfeel and show the high score for description of spicy, floral, and raspberry. So, concluding the road of my research activity, we can say for the activity 1 and activity 2 that oak surface adhered Starmarella bacchillaris cells could be a good strategy to improve the volatile sensory profile of Montepulciano and Trebbiano d'Abruzzo wine, and could be a good strategy to propose to wine sector in order to obtain novel style of wine. Regarding the activity 3 and activity 4 related to biofilm detached cell, we can say that wines obtained with biofilm detached cell shows an increase of red tint than those obtained with planktonic cell, and the different lifestyle of Starmarella bacchillaris can generate wines with different aroma characteristics. Regarding the future perspective, could be interesting find the difference in gene expression between planktonic and sessile cell, in particular biofilm detached cell, in order to better understand how works this metabolic shift and study the color difference, maybe with the evaluation of cell lifestyle difference in cell wall. Thank you for attention, and
Thank you very much. Any questions from the audience? Nobody's curious which kind of Montepulciano is. Okay, thank you very much. Let's move to the other speaker. And we have Eugelio Fabiola from Contaminants to Geographical Markers, the case of quinolizidine alkaloids in Lupinus albus. She's online because she was presenting another talk in an international conference. Hello.
Good morning, everyone. I will present you the window now. Okay. Can you see the presentation? Yes. Okay. Good morning, everyone. Thank you for the presentation. And today I will talk about a project on which I worked during the last PhD year. Let me introduce you, first of all, the alkaloids that are heterocyclic compounds containing nitrogen. They are secondary metabolites that originate from amino acids, and they are present in plants, in which they perform different biological functions. For example, they act as a defense mechanism for the plants, conferring their typical bitter taste. Different classes of alkaloids can be found in nature, but today we will focus on quinolizidine alkaloids that are the most abundant compounds that are present in the leguminosa lupins, and they can be found in different forms such as bicyclic, tricyclic, and tetracyclic derivatives. So the problem is that these alkaloids are very toxic and could lead to adverse effects on both animals and humans' health. For this reason, in order to make the lupins edible, it's necessary that lupins are subjected to specific washing and embitterings processes with the aim to decrease the levels of alkaloids, below the maximum levels allowed from the international guidelines. So these alkaloids, is necessary to inform you that the levels and the type of alkaloids that are contained in lupins is very variable, and it is depending on different factors such as geographical origin, lupin species, soil composition, stress factors, and so on. For this reason, due to its high variability, their content, our proposal is to use them as biomarkers for food traceability. These here are presented the most known alkaloid structures that can be contained in different lupin species. But in particular, I want to show you the most representative alkaloids, in particular the lupanin of Lupinus albus cell, that is the object of our work. Currently, in the literature, there are different works about the study on the exploration of alkaloid profiles of different lupin species, but most of them are performed in GCMS. There is no any study about that in LCMS, and no fragmentation studies of each alkaloidic structure in CID. For this reason, the aim of this work was to develop a semi-targeted approach by LC, MRM, IDA-EPI for the putative determination of all the alkaloids in lupin samples to have a complete characterization. So the aim was to apply this method on Lupinus albus cell samples of different geographical origin and apply multivariate approaches analysis for the discrimination and classification. So this is the workflow that of the method that we developed, that is characterized by sampling of lupins, an extraction step, a cleanup by solid phase extraction, analysis by HPLC coupled to a hybrid triple quadruple linear ion trap mass spectrometry, acquisition in MRM IDAEPI, and then application of multivariate approaches on our data to consider to see if these analytes are able to see if these models are able to classify our samples according to the content in alkaloids. So this is the procedure, the analytical procedure that we applied on this method that we previously validated in another work for the targeted determination of alkaloids in lupins. In particular, we focused on the tuning of a cleanup step by solid phase extraction with the aim to reduce the matrix effect and obtain a selective recovery of all the analytes thanks to a reverse phased polymeric sorbent StratXL. So, but now, considering the limited availability of alkaloidic reference standards, we built a semi-targeted method with the aim to have a complete characterization of the alkaloid profile in lupins at putative levels. And so, for this reason, a bibliographic research of all the alkaloids contained in non-lupin species was performed. Then, an in silico spectra prediction for all of them by the software CFMAD was performed to obtain the creation of a database. This database was then used during the semi-targeted analysis, during the survey scanning multiple reaction monitoring, with the aim to search during the analysis in our samples all of these structures with the aim to obtain, by the activation of AP experiments, a fragmentation pattern from each of them. So the final result is the detection of each analyte and the the generation of an MS spectrum. So these are the instrumental parameters that were tuned during the building of the method as regards both the chromatographic separation and the mass spectrometry detection analysis. Once the method was built, it was applied on Lupinus albocell samples deriving from different Italian regions highlighted in the image. And 27 alkaloids were identified thanks to this analysis. So 6 of them were identified in targeted mode by multiple reaction monitoring thanks to the use of reference standards. While here are reported some of the 21 phenolizidine alkaloids identified thanks to the semi-targeted approach at putative level. As an example of the identification, here we have a chromatographic peak detected in multiple reaction monitoring. According to the information obtained by the literature and in silico, this could belong to the angelloxylupanin, a derivative, an ester derivative of lupanin, because the lupanin is the most abundant alkaloid in Lupinus albus, and thanks to, by means the activation of experiments, the MS spectrum was obtained. So, thanks to X spectrum, we can proceed to the identification of the molecule. How? We matched the spectrum both with the predicted spectrum obtained by the CFMAD software, but also with the spectrum of the reference standards lupanin. In fact, many fragments in common we found, and we also hypothesized the structures, so we can putatively confirm that the precursor ion 347 could really correspond to angelloxylupanin, and this kind of identification was then performed for all the analytes detected. After the identification, with the aim to consider these alkaloids as biomarkers for traceability, two approaches of multivariate analysis were performed. First of all, an unsupervised hierarchical clustering was applied. We can see from the heat map that we have 3 clusters. The first one on the left is the better for the discrimination of the regions, while the second the ones at the center of the heat map will discriminate only Abruzzo Campania from Puglia, Lazio, while the third one is the most variable and characterized by non-discrimination of region. The thing that I want to show you is that the third cluster is mostly represented by the alkaloids detected in targeted, while the first one, that is better for discrimination, is mainly represented by the putative identified alkaloids during the semi-targeted approach. What does it mean? It introduced us that for a good classification and discrimination of the samples is necessary to obtain a complete analysis of alkaloidic profile. So for this reason, we then applied a supervised approach in PLSDA cross-validation, at first only on the 6 target alkaloids, but we didn't obtain satisfactory results. After that, we applied the same model on all the 27 alkaloids, obtaining satisfactory results in terms of sensitivity, specificity, and so on. But also we can observe in the table the synergistic contribution of the putatively identified alkaloids thanks to the semi-targeted approach in the classification of the samples originating from different regions. For this reason, we can confirm the possibility of use these alkaloids as biomarkers for traceability. So in conclusion, a semi-targeted approach by LCMRM IDAP was developed. 21 alkaloids were putatively identified. The samples were successfully classified according to their geographical origin thanks to a supervised PLSDA, and so that confirmed the possibility of use these alkaloids as markers for geographical origin. And the work was recently published on Food Chemistry X. I want to thank you, my analytical chemistry group of Teramo University, and all of you for your attention.
Thank you very much. As usual, any question? Come on. Oh, finally. Please. Come here.
For somebody who knows nothing about the subject area, can you hear me? Can you hear me?
So and so.
Wait a second.
I come up to the microphone.
Yeah.
I'm curious as to, because I'm interested in traceability, but did you have the opportunity to investigate the year-to-year variation because of climate or different climates? Will that compound your conclusions relating to ability to
we have the possibility.
No, again.
I am interested in your traceability and regional variation, but I am wondering, will annual variation in climate, I mean different rainfall or sunshine, will that complicate the possibility to be able to identify the regions, the geographic origin of your samples?
Actually, currently, we only consider the geographical origin according to the samples that we had in our stock. But it's possible that climate and the stress factors could contribute to ulterior variability on their content. And so it could be interesting to study if also in this case applying different models of statistical multivariate analysis is possible to classify these samples or not. But we can consider that in these different regions that we consider, there is a little difference in terms of climate conditions and also soil conditions. So it could be interesting to deepen this aspect.
Okay. Any other question?
Sorry.
Please. Something else? Fabiola. Did I stop you? Devi dire qualche altra cosa?
No.
Okay, sorry. Any other question? Okay, we move forward to another speaker that is the same conference. And actually, before that, I would like to thank Professor Schirone, that is online, she's the one that organized mostly this annual meeting. She had a problem, so you can see her with the M on the screen. So thank you very much, Maria, for your work. Okay, let's move forward. And we have Silene Fiori, paper-based sensor based on biochar sensing film for bioactive compounds determination in agri-food byproducts. Silene, the floor is yours.
Yes, good morning. Can you see the presentation?
Yes.
Okay. So, good morning, everyone. Today, I will present you a work based on the development of a biochar paper sensor following the principle of circular economy and sustainability. Unlike the previous linear economy, the circular economy tends to reuse waste and byproduct in order to reinsert them into the production cycle. So, following this view, we develop a sensor based from waste of paper industry. In particular, we use biochar as sensing element obtained from lignin byproduct and recycled paper used as substrate for sensor. This biochar paper sensor was then applied for the valorization of plant-based waste and byproduct rich in bioactive phenolic compounds. Nowadays, paper is widely used for fabrication of analytical device thanks to its properties that can be further improved with integration with other nanomaterials. Between these nanomaterials, biochar is considered as sustainable and, to the best of our knowledge, only to work report its integration with paper for electrochemical sensor. Biochar is a carbonaceous material that, thanks to its properties such as large surface area and good stability, can be applied in a big number of fields, for example, as soil amendment, adsorbent material, or for supercapacitor. Biochar is obtained from carbon-rich biomass, and in particular, the one used in this work is obtained from black liquor, that is a lignin byproduct of paper production. Biochar is obtained with pyrolysis process performed at controlled temperature and atmosphere, and depending on the parameter used and the biomass, we can have biochar with different structure and properties. In this case, biochar is obtained from eucalyptus scrap and present a nanofibrillar structure. These nanofiber in bark form are not dispersed and agglomerated, so biochar is subjected to ultrasound-assisted liquid phase exfoliation in water in the presence of sodium cholate, that is a natural stabilizing agent. The dispersion obtained in this way is then filtered onto cellulosic paper, and the biochar film is cut with a CO2 laser in order to obtain the working electrode. Then we choose a substrate that is paper obtained entirely from recycled cellulose, onto which contact and counter electrode were printed with a carbon ink using an adhesive stencil printer. Then, after curing of the ink, the reference electrode were printed with a silver ink with the same method. Then sensor was completed, integrated the working electrode using an adhesive spray applied on the back of our electrode, and the insulation was performed with nail polish. To obtain the better analytical performance, the amount of biochar has been optimized, and this optimization was performed first with the classic inner sphere reductor of ferroferrisanide in cyclic voltammetry and impedance spectroscopy. In this case, we can see as the electrochemical parameters improve with the amount of conductive material. Since biochar is also used for supercapacitor, we evaluated the capacitive current with cyclic voltammetry of potassium hydroxide, Also, capacitive current increased with the amount of biochar. So we need to find a compromise between a not too high capacitive current that can hide signal of the analyte and a good Faraday current. For this reason, we excluded the highest and the lowest amount of biochar. Then optimization was concluded with differential pulse voltammetry of 4 phenolic compounds that are often found in waste and byproduct that are caffeic acid, chlorogenic acid, oleuropein, and catechin. This was performed in differential pulse voltammetry and the current intensity was evaluated. In this way, we selected 8 milligram of biochar as optimal amount. We also characterized the optimal sensor with the 4 analyte in cyclic voltammetry, comparing result with a commercial screen-printed electrode. And we can see an improvement in the performance both for current intensity and for reversibility. Then we will calibration curve with increasing amount of the 4 analyte in differential pulse voltammetry. Since the aim is to have a circular process, we applied this sensor for the determination of polyphenolic compounds in plant-based waste and byproduct. In particular, we had 2 food samplers from olive leaves and artichoke leaves, respectively for oleuropein and chlorogenic acid, and 3 food waste that are cocoa husk, coffee husk, and exhausted coffee pod for catechin and caffeic acid. After extraction, the quantification was performed with the standard addition method. Here in the table I report the result, and the biochar sensor proved to be sensitive and with good reproducibility and quantitative recoveries. Quantification was also proved compared with classic photometric methods that are falling chocolate, golden nanoparticle assay, and ABTS. In this case, results do not coincide numerically because this quantification is based on different principles for the different method, but we have the same trend with the highest polyphenol content in olive leaves food sampler and the lowest content in cocoa husk. Then we prove selectivity of our sensor, performing DPV in presence of different compounds such as ions, sugar, organic acid, and polyphenols that are often found in this sample. In this condition, with the same dilution used for our analytes, we don't have any interference from this compound, with the exception of picumeric acid and syringic acid that show some signal, but these don't overlap with the one of our analyte. Then, as last step, we also evaluate the sustainability of the proposed sensor with the white analytical chemistry, that is a concept introduced a few years ago and expand the classical concept of green chemistry also to a red and blue chemistry. Red chemistry is about analytical efficiency, so it considers parameters such as limit of detection, the selectivity, the number of applications of the method. Chemistry is about socioeconomic efficiency, so about cost, energy consumption, and instrumental requirements. And then we have the classic green chemistry about environmental sustainability. The combination of these three will give rise to a white chemistry. To have a whiteness of the reaction, we give a score from 0, that is the worst, sustainability to 100, that is the best, to each color. And then the combination of this color gives a whiteness percentage. We then compared our sensor with photometric and chromatographic method for polyphenol determination. As we can see, for red score, we have a less point for our sensor compared with chromatographic method because we lack in selectivity and sensitivity, but we have good score for blue because our sensor is cheaper, consume less solvent, do not require particular instrument, and is easy to use. So in conclusion, here I propose a biochar paper sensor obtained entirely from recycled materials from paper industry, and we try to minimize the number of steps, the use of solvent, and the production of waste. We also apply this sensor for valorizing food waste and byproduct. And sensor shows to be selective, sensitive, and gives good recoveries. So I thanks my research group in the University of Teramo, the PON fund that allowed me to have this doctorate, and thanks you all for your attention.
Thank you very much. Also here, any question from the audience? Okay. I have a question. Can you hear me? Hello?
Yes.
Okay. Sorry, just a question. Clearly, it's quite interesting, but in terms of costs, for the classic total phenolic, that it's quite easy despite it has a lot of interference, ascorbic acid, reducing compounds, and so on. Is it costs less? How is compared to the total phenolics? I mean, I understand that it's more sensitive and so on, but clearly the Falling Chocolate is the method that's been used for years for phenolics. And the other question is it a method that is focused just on phenolics in terms of bioactive? Because, you know, bioactive means that somehow you have to calculate the bioactivity, so you don't know if it works or not. And, but it seems that it's really focused on phenolics. So you already did a determination of the bioactivity.
Okay. Regarding the first question, we evaluated cost not only of, for example, reagent, but also instrument. So, for example, for Falling Chocolate, we also need an instrument to read measure. And obviously, in this way, costs are higher than, for example, our portable sensor. Regarding this second question, in this case, we don't evaluate the bioactivity, but this is just the amount of phenolic compounds present. Because obviously, with electrochemical method, we need electroactive compounds.
Yeah. Okay. Thank you very much. Any question from the audience? Okay. Thank you very much, Stan. And let's move to the last but not the least speaker, Trasmundi Flaviano, mitigation of salt stress in durum wheat and horticultural crops through PGPR inoculation potential for sustainable agriculture productivity. Please.
Okay. Okay. Good morning to everyone. Today is a pleasure to be here, and thank you for a presentation. Today I want to talk about my PhD project, mitigation of salt stress in durum wheat and horticultural crops through inoculation of PGPR. The main challenge of the agriculture are named climate change. The fifth report of IPCC highlights the vulnerability of agriculture to climate change. So it's most important to focus our attention to then can be the future of the agriculture. If we imagine the forecast to 2050, the temperature will increase in global average for a minimum of 2 degrees or maximum of 5 degrees. And this have a lot of consequences, a lot of damage all over the world. For first, here I listed few motivations, but we can mention the rise of demand growth or the lack of some resource, especially water, the rain for the alteration of the seasonal trend of precipitation, and the extreme event, the increased frequency and the intensity of floods and drought. So, what are the solutions of climate change? Here, I'm not here to resolve this problem, or the climate change maybe will be not resolved, but we can give a hand to the agriculture or the nature or the soil. I focused my study on I focused my PhD project on study of plant growth promoting rhizobacteria. Usually, when we talk about microorganisms, we don't mention the microorganism of soil. Maybe we think about clinical microbiology or human microbiology. but never about the soil. The soil is most important and is important to maintain the health of the soil. The plant growth promoting rhizobacteria, here we can see only few of the capability that can do to the soil, the nutrient to the soil and to the plant. The nutrient uptake, the ionic balance, and antioxidant system boost. And here is the survey. So we know that have microorganisms on soil, but we know also that we have a very, very good variability in soils. So the difficulty is to establish a stable systems of microbiome in soil, especially in my case of PGPR, because we have a long-term variability, a spatial heterogeneity, and complex interaction between plants, soils, and microbiome. And another aspect that we can consider is that this specific condition that may not generalized in other agricultural systems. After that, I start with my project. I choose to isolate PGPR in Crea, in Monte Polo del Tronto, in Ascoli Piceno, in Marche region. First, the idea is to isolate potential PGPR. We isolate in 2 vegetable crops, cucurbitaceae and tomato, managed according to the no-tillage method. But why we choose no-tillage? Why we choose organic? The no-tillage method is associated with less soil disturbance, so maybe we can have more biodiversity. The choice of organic system is in the logic of reducing the use of chemical input, because if we think all of the damage that the chemical input we have with conventional agriculture, we can translate also the damage about the climate change. About the choice of tomato or cucurbitaceae, we can see that cucurbitaceae have extensive and particularly root systems that tomato. The tomato are usually used in literature for isolate this type of bacteria. But about cucurbitaceae have the system, the radish, that can have a unique ecological need for strains, and in this case, PGPR. The first step, or the first part of my work, is considered to also to see the difference about the total bacterial concentration in each of soil, organic cuc NT and organic tomato NT. We can see, as I said previously, that in organico cucurbitaceae no tillage we have measure a significantly difference in front organic tom NT. After that, preliminary selection and screening for PGPR activity. Okay, we isolate a total of 148 bacteria. We're selected and firstly screened for their capability to characterize a PGPR activity. Production of pheromones such as biostimulants, indolacetic acid, phosphorus solubilization, production of siderophores, and antagonistic activity against pathogen. So we start from 148 bacteria, but only 28 are characterized for PGPR activity. We did the molecular identification, and we can see in this table that we have a lot of bacillus. We did also the differentiation through this PCA about the organic tomato NT and organico cucurbitaceae NT. In organico cucurbitaceae NT, according also with the literature, we have most presence of bacillus species. And the bacillus species usually are the best PGPR. Okay, so we isolated PGPR, we identified, and now we want to know how this plant growth promoting works. About the analysis of indole acetic acid production, siderophores production, and phosphate solubilization. We start now with siderophores productions. Why is good siderophores production? Because is an alternative to fight the molds in pre and in post-harvest. We can see here that Bacillus allotolerans, A, B, and C, this letter mean the strain, and Bacillus pseudomycoides and Bacillus veleziensis produce higher 18% of siderophores. These results can be translated in this biocontrol activity. We have here Fusarium oxysporum and Alternaria alternata. These are the major contaminants in pre and in post-harvest and are responsible to produce mycotoxin. So, Bacillus allotolerans A, B, and Bacillus allotolerans C and veleziensis, we can see the inhibition allo. Here, and Bacillus pseudomycoides have the capability to grow under the molds. So, the results about siderophores production are justified. But we have also to do another analysis about maybe the production or some volatile compounds or some antibiotics. After that, after the biocontrol activity, we asking about, okay, this PGPR have good biocontrol activity and agonistic activity, but what are the effects on plants? Specifically in wheat sprouts. We use salinity stress, because salinity stress is the major, or salinity or also water stress is the major that is a cause of caused by climate change. Here we can see a plate with seeds at different concentration of salts. Okay, about the control, we can see at 200 millimolar of NaCl, we have a reduction of germination performance. But when we inoculate with Bacillus altitudinis, a PGPR isolate, our PGPR isolate, at different salt concentration, we can see a raise of PGPR, a raise of germination performance. This result also about Parthenobacter nitroguaiacaulis at a different concentration. Another things, okay, we have a good germination performance, but we choose to compare our germination performance with a commercial biostimulants. We choose Micosat F. Micosat F, as we can see in this image, have a very good germination performance. But with our bacteria, we don't have a most performance by Micosat F, but we have the same. And this is our goal, have isolate our bacteria and have the same characteristic of a commercial biostimulant. Okay, after that, we can translate these results in plates with the analysis. We did the phosphate solubilization and indole acetic acid production. As I said previously, indole acetic acid is a biostimulant, and Bacillus altitudinis can produce higher 20 ppm, and is good also about the phosphate solubilization. Can have the phosphate available to the plants. Okay, other tips. Okay, we concentrate our attention also to the stability of our PGPR to salinity condition. This is a firstly screening, very fast screening that we can see grow, no grow of our PGPR. But We applied a Gompertz model. We can see that Bacillus allotolerans at different condition of salts until 400 millimolar can grow perfectly. Bacillus allotolerans, Parthenobacter nitroguaiacolis, Peribacillus frigotolerans, and here we have a Mycosat F. And another time, the important thing is that our microorganism growth well like Mycosat F. Okay, here I resume that I said. And here, another question. Okay, our bacteria have a plant growth promoting ability, but we worked also with sprouts. And we have to think if our sprouts can be commerciable with an upgrade of total flavonoids and phenols, chlorophylls and carotenoids, and antioxidant activity. We can see here that when inoculated our microorganism with the sprouts, we have a higher concentration of total flavonoids contents and total phenolic content. This can be an opportunity to have a commercial sprouts that we have a higher concentration of total flavonoids. Antioxidant activity, we have the same trend, 0, 100, 200 millimolar. We have a decrease of antioxidant activity confirmed about ABTS and FRAP. And chlorophylls and carotenoids that have a repercussion about the growth. Higher concentration at 0 millimolar of salts. Here, the same about chlorophyll B and carotenoids. About the conclusion, I run from the conclusion. So, summary. We identified PGPR, inorganic no-tillage systems. We identified and characterized a total of 28 bacteria. Selection by biostimulant capacity, biocontrol capacity, protection from salt stress, and increasing the quality of wheat sprouts. Now, what is the survey? The survey is to develop a PGPR consortia that will be stable in different agriculture systems. Because this is the survey, and this is the difficulty of this work. Because one thing is to work in laboratory, one thing is to work in open field. We have a lot of discrimination. So we hope to have a PGPR consortium for challenge climate change. Thank you for the attention.
Thank you very much. Any further question?
Thank you.
Okay, too shy in audience. Oh, please, thanks.
Sì, yes. Lurea bertani, yes. Thank you.
Okay, so now we have coffee break in that room and also poster session. So I invite you to give a look to the poster. We come back around 12:30 for final remarks and, of course, awards for the prize. Thank you.
Pensavo di voti per partire qua vicino, però
Qua? Sì, ma lo senti, è meglio. Me lo sento.
Ma io fino all'altro giorno ti ho visto qua e ora?
è partita.
Ora sei partita proprio sul più bello?
Sì, sì, tutto bellissimo.
Ok, senti, tu riesci a condividere il poster?
Devo cercare, sì.
Così facciamo prima, se no di là c'è un gran casino.
Sì, rischi di non sentire nulla.
Ok. Sai che non so se a questo punto riesco.
No dai, non ti preoccupare, descrivimi il lavoro a voce.
Aspetta, che se no te lo
Io adesso
lo apro io.
Perché da me non riesco. Riesce a farlo lei proprio? Ok.
Aspetta un attimo.
No, non c'è fino al secondo, sono due primi.
Scusami, sì, per il primo e il secondo anno.
Lo vedi?
Sì, sì, sì.
Chiara?
Oh,
Sì, sì.
Perfetto, vai.
Sono tutti presenti. Allora, questo progetto si concentra principalmente sulla nutrizione e sullo sviluppo del nuovo indice infiammatorio nutrizionale o NIA. L'immunonutrizione guarda a migliorare la salute identificando schemi dietetici che possono mitigare le condizioni patologiche legate al sistema immunitario e modulare l'infiammazione. In passato è stato già sviluppato un indice, però questo indice è un primo tentativo, quindi presenta delle problematiche, quindi non è eterogeneo con i dati e anche non è basato su dati sperimentali. Per questo l'obiettivo di questo progetto è sviluppare un indice infiammatorio basato su un modello solido sperimentale per valutare i ruoli antinfiammatori e proinfiammatori dei cibi e di ricette. Quindi abbiamo selezionato alimenti e ricette che rappresentano le abitudini italiane e noi le processiamo mediante un protocollo in vitro, quindi le digeriamo mediante l'InfoGest, che è un protocollo il quale minima le 3 fasi della digestione, quindi la fase orale, la fase stomacale e la fase intestinale. Dopodiché abbiamo impostato un modello cellulare che è un'agricoltura utilizzando delle cellule H2 e HP29 MTX e MTX e 12 e le PHP1 per simulare l'intestino in condizioni sane e infiammate. Ogni alimento è valutato in base al profilo del rilascio di citochine. Ad esempio, in questo poster abbiamo testato il digerito d'arancia. Come si può vedere, abbiamo trovato solo un risultato significativo nella concentrazione più alta quando abbiamo valutato il TNF-alfa, che è una citochina proinfiammatoria. Questi risultati supportano gli obiettivi del progetto che esplora il ruolo della dieta nella modulazione della risposta infiammatoria e quindi il passo successivo è completare il test di tutti gli alimenti, anche dei cibi e delle ricette e sviluppare un algoritmo per poter mediante un numero valutare sia anche queste ricette e alimenti, però anche dei modelli alimentari già fatti come la dieta mediterranea e altri tipi di dieta, fornendo un riferimento per schemi alimentari sani. Grazie. cereali, di carne, di pesce e uova e di anche di prodotti lattiero-caseari e ovviamente frutta e verdura.
Ok, quindi per avere insomma...
Sì.
Ho capito, va bene, grazie.
Grazie a voi.
Ok, grazie.
Grazie.
Allora, dai, chiamiamo la Spirone. C'è questa cosa, allora, Paolini, allora i conti sono fatti bene.
Sì,
A me è piaciuto
60
lui.
in totale.
A me è piaciuto.
Lui è 52 più 8.
Cioè, Paolini, scusa, Rossetti. Allora.
52 più 8, se 8 gli volete dare, se no...
No, a me la presentazione
era scritto bene.
mi è piaciuta
La
un sacco.
presentazione è stato l'unico
Sì,
che è riuscito
sì,
nei tempi stabiliti
sì.
a
Poi è
fare
chiaro.
l'escursus di tutto quello che ha fatto, quindi secondo me è stato...
Maria, eccoci. Allora, sto qua con Lilia. Per poster, vabbè, è chiaro, quello che abbiamo al massimo. Va bene. Allora, dunque, abbiamo rifatti i conti e i conti erano quelli, cioè, quindi teoricamente sono conti niente, cioè nel senso che, come si chiama qui? Rossetti come punteggio dei lavori è il primo. Poi gli manca la parte award e la comunicazione, magari quello poi gli si dice. Io devo dire, non so se hai sentito le presentazioni, che è stato veramente bravo, ma bravo. E quindi a questo punto, visto che la valutazione numerica, che però non è che dobbiamo attenerci per forza a quello, quella era quella del premio Reggiani, no? E effettivamente lì abbiamo dato un sacco di punti alle pubblicazioni, però effettivamente questa ha fatto 13 lavori, cioè sono tanti, sono veramente tanti. Manca un po', cioè sui premi, quello vabbè dipende anche da chi è il tuo, no, tu per mille cose. La parte che manca è quella della comunicazione, che qua è un po' strano perché insomma si parla di vino, quindi, però ecco, lui ha fatto veramente un'ottima presentazione. Quindi io voto lui, cioè non so voi.
Cosa pensate invece di Eugenio? Perché l'altra, secondo quella che era la
Sì.
critica che abbiamo fatto, ci sarebbe con 58 ci sarebbe la Fabiola.
Sì, seconda però.
Eugenio.
Sì, infatti il punto è tra loro due.
Io ho 58, se 52 era Rossetti, 58 è l'Eugenio.
No, 52, però dopo...
No, l'hai fatta, aspetta. No.
Allora, lì
Tira
c'era
fuori.
la valutazione, quella sull'abstract, no?
Sì, allora lì il punto era che...
Poi c'è da sommare a tutto il resto.
Sì,
Quindi
allora il
c'era-
punto era, qui veniva, quella l'ultima che ci ha mandato Lilia, primo era Rossetti. Però poi tu hai detto che hai fatto il calcolo, però Lilia mi dice che nella sua valutazione i 3 capitoli non c'erano.
No, cioè io...
Ok.
Allora, sono 13 pubblicazioni che dove Rossetti è il primo nome
In due.
soltanto in due.
Sì.
E poi c'è
9
unità per il
Uno.
punteggio in uno.
Sì.
e fa 52 più 8
Quindi sono 52, giusto?
più 8
Quanto ti viene nelle pubblicazioni?
sì, è vero.
Scusate, a me viene 39, 40, 41, 42, 43, 4, 5, 6, 7, 8, 9, 10, 11, 12. Sì, 52.
Ok, 52, quindi il totale suo è 65.
Vabbè, sommati giustamente.
E il totale dell'altro è 58, perché gli abbiamo dato tutti 8, io quanto gli ho dato? No, 8, 7, 8, 8, 7. Questa avete dato tutto quello, 0 per il premio, invece l'altra c'è il premio. Per la distribuzione c'ha lei l'altro, c'ha molto di più, c'ha 10. Poi il nostro voto di commissione abbiamo dato 5 a tutti praticamente.
Abbiamo dato 5 a tutti, sì, tu dicevi.
Siamo stati larghi?
Sì,
No, scherzo. Sì, sono stati bravi.
Tu dicevi non premiamo le pubblicazioni, premiamo anche la questione della disseminazione.
Sì, io pensavo quello, no? Però è chiaro che pure la presentazione ha una sua importanza, poi vedendolo insieme va bene pure così, insomma, magari si dice dalla presentazione. Allora, quindi adesso facciamo una prevenzione, allora, dai. Grazie, ciao. Allora. Sì.
Chiara Fittetta
Sì, qua ci sono i fogli poi da tutti i periodi per dare la premiazione. In realtà qua non c'è scritto c'è tipo il nome da scrivere, quindi non so, o lo scriviamo al lato, glielo diamo e basta così.
No, io lo scrive Mauro.
Qua c'è l'oral e qua c'è il post. Sì, no, dico nel senso che non c'è uno spazio per il nome, adesso magari chiedo un attimo a Luana, ma non credo che sia stato pensato. guarda, dice tanto lei poi comunica a Mario per il pagamento, però se vuole, cioè, se voglio scrivere il nome giusto per farlo vedere. Sì, esatto, ho detto, a me fa rimanere loro.
Però
Sì, sì.
bisogna firmare Mauro.
Vabbè, tanto scrivi tu che scrivi lei.
Allora, yes.
Okay, so welcome to the last part of this morning. I would say also the most pleasant. So, where are? Okay. So, as you know, we have some winners today for the best poster presentation. We confirm the award to the PhD student Chiara Trippetta. Okay, unfortunately,
She's in Cambridge.
she's in Cambridge, so she.
Make a picture with Donato.
Una foto con te.
Okay.
Dottorando un po' cresciuto.
Okay, and as concerned the PhD students of the third year, the winner for the best oral presentation is Alessio Rossetti. Okay.
Just a brief explanation. It's not that we decided that he win because we like him. There is a clear schematic assessment of different aspects of his path. That is the same that we utilize for the Regeni Prize. So it's involved in different aspects of the 3 years. The publication, you have different points if you are first or last author, if your paper is on Q1 journal, if it's only international journal can be assessed, and so on. And clearly also the other 2 aspects of the PhD project that are related to dissemination, scientific dissemination, intermission, and also poster prizes awards. So the mix of all these indicators together with our assessment in terms of listening to the presentation, and I have to say that his presentation was very clear. First of all, it was on time. It was probably the only one that was on time, and that's really important. It was extremely clear and very formative also for someone like me that didn't know much about it. So that's the reason why we selected him as a winner. For the poster, clearly we have first and second year. It's extremely difficult that we give a prize to the first year, because you are just presenting the project. So we don't have any kind of marker or assessment to decide that will be important. So we follow the same approach, just on the abstract in this case, and on the discussion. And at the end was the winner, I'm sorry, is from nutrition. I mean, I have to say that we have a maximum of 8 points.
I was the only one of the other two that gave less points to her. So just forgive my integrity under this point of view. And now, final remarks. I think it was fine. We are, all of you are here. I'm very sorry about the fact that tutor didn't show up, except 2-3 tutors. And that's something that we have to work on. We need more presence by the tutor. So try to push them. I will do it. You know that I will do it. So we will see next year for the annual meeting, but also for the PhD Food Arena. We have some news about the PhD Food Arena. We are trying to do it in synergy with Milano and Parma University that have a similar program in PhD, food science, and probably in order to avoid too many people, just for the third year PhD student. We'll see. Will come news about it. For now, I will thank you, all of you. I will thank Lilia and Maria for the big effort they put in this organization, and I wish you a good day.